Good morning, all. We will begin in a few minutes just to give the other participants time to log into the webinar. So we will be beginning at 11:05 sharp. Thank you so much for your patience. Good morning once again, everyone. Welcome to today's webinar titled The Bahamas Coral Innovation Hub, Improving Coral Reef Restoration Through Research, Outreach, and Education. So our speaker for today is Natalia Hurtado, who is the project manager of the Bahamas Coral Innovation Hub, located at the Cape Eleuthera Institute at the Island School. So before we get started, just a few housekeeping rules. Please note that this webinar is being recorded. And just a reminder to keep your microphone muted during the session. Afterwards, there will be a Q&A session. You can add any questions into the chat, or you will have the option to raise your hand and ask your question. And an audience feedback form will be shared in the chat before this webinar ends today, and we would really appreciate you guys filling out that form. So without any further ado, I will now welcome our speaker to begin their presentation. Natalia?
Thank you so much, Neil. I hope you can, I think I lost it. Okay, can you see my screen?
Yes.
Perfect. Good morning, everyone, and thank you so much for being here today. So I'm just gonna talk about the work that we have been doing here. I'm not really sure if you are familiar with the Bahamas. Probably you heard about that before, like a really cool destination just to go on vacation, just to enjoy the beach. We have millions of tourists coming here every year. We have a lot of people coming here on a daily basis. And probably it's not just to enjoy the beach, maybe just to see the sharks, just to relax. But the reality is that coral reefs are extremely important for all of us, just not for the ecology, economy, but also the culture of the Bahamas. We are located in the Northern Caribbean. And the Bahamas, we have more than 700 islands and keys. So this is like a little map where you can see where coral reefs are located in the Bahamas. We have actually a really big barrier reef in Andros. We don't have a lot of coral reefs in Eleuthera, but we still have some coral reefs around. And as you might know, coral reefs, they're super important for coastal protection. They provide habitat for marine life. They're important food resource, and they have a really, really huge economic value. So the Perry Institute for Marine Science, we preserve coral reefs, coastal habitats, also fisheries, and we also work with local communities and implement sustainable solutions. At the Perry Institute for Marine Science, we work with fisheries, as I mentioned, coastal communities, just communities, coral reefs, and for coral reefs, we do coral reef assessments. We also study diseases, and we have a really huge restoration program, which is, it was created, the Reef Rescue Network. And then at the Cape Eleuthera Institute, we promote conservation of marine and coastal ecosystems. We host a lot of visiting scientists. We support education. And then we have different research groups working with different marine animals. We have some group working with lobsters, with sharks. We work with oceans and climate, food security, resource management, fish physiology, and of course, we also work with coral reefs. So since these two institutions work with coral reefs, the two institutions came together, and with the support of the Nature Conservancy, the Bahamas Coral Innovation Hub was created. We are located in Eleuthera. That's one of the islands of the Bahamas. We are in South Eleuthera. We are a center for development, implementation, and dissemination of scalable coral restoration techniques just to help to counteract coral reef decline. We have land and ocean-based nurseries. We also work with larval propagation. We do monitoring. We also work with important grazers like long-spine sea urchins, and we do a lot of outreach and education. So here in the Bahamas, of course, we're also part of the Caribbean, so we have a lot of different coral species. We have the most common coral species than other places, but with all this difference, we also need to apply different techniques, and we also face different problems. So same than other places in the Caribbean, we also were affected by white band disease in the 80s, where most of our populations declined. It was a mass mortality, and in some locations, these acroporids are locally extinct, so it's not common to see acroporids anymore. And that's actually one of the reasons why most of the coral restoration projects started in the Caribbean, not just here, but in other places. It was mainly focused on trying to bring these kind of species back after these mass mortalities in the 80s. But besides this kind of disease, we also face the increase of heat waves. So in 2023, we have a massive bleaching event. This was just not for the Bahamas, it was also for other places in the Caribbean. Then we also have a lot of, we have like a huge impact of diseases. We also start facing a lot of problems with the stony coral tissue loss disease. And in this picture, you can see how after the coral get infected, after 2 months, this coral just can be completely dead and covered in algae and sediment. So this disease was reported in 2019 here in Bahamas, started in the States. But of course, this rapid mortality is just creating a lot of problem for us. We are doing treatments using amoxicillin just to try to stop the disease in some of these corals. And we also have been recording these bleaching patterns and which species have been affected after these bleaching events. But after mass mortalities and after bleaching events, we also have massive mortalities, mainly because the corals are more susceptible. We have diseases, outbreaks. And right now, the reefs are not really in good shape. So we have a lot of important species that they've been dying, or actually is just really hard to find on the reefs nowadays. For example, brain corals were heavily affected, not just by bleaching event, but also for stony coral tissue loss disease. So then just give us like a lot of challenges for our coral restoration efforts. So here at the Bahamas Coral Innovation Hub, we use the different reproductive strategies of corals. So one that we use, and that's just like commonly known for other places, is asexual reproduction. So we use fragmentation, where just a little piece of just a piece of the coral breaks, and then that piece is just gonna grow again and just gonna form a bigger colony again. And then the budding just for increase the size of the coral. So this is a technique that we use to increase abundance of different coral species. We use mid-water nurseries. So we just cut a little piece of the coral, like around 5 centimeters. Then we put it in a stable place, which is our mid-water nurseries. And this process is known as fragmentation. And these small fragments, we normally raise them in ocean or land-based nurseries. And then we also applied microfragmentation. That technique was started in the States, and we started applying this technique here, especially for boulder corals. So the fragmentation is mostly for the branching corals, and then the microfragmentation just for boulder coral species. So here's like a picture of the before and after of how our lab used to look. So at the beginning, it was just like a space with a couple of tables, a couple of containers, just like 2 raceways just to try to conduct some experiments. But then with the support of TNC, we were able to improve capacity. We were able just to build a lab where we can control different conditions just to try to boost and help with coral growth. And then also we built an outdoor system. So same, it was just like a space in the middle of the bush. So what we did is just we built an outdoor system just to be able to start some of our efforts in different kind of conditions. So right now we have a full functional land-based nursery, like with indoor and outdoor tanks. We also have and ocean-based nursery. So we are part of the Reef Rescue Network. This Reef Rescue Network, as I mentioned before, was created by the Perry Institute for Marine Science. And then for this specific technique for fragmentation, so we use these midwater structures, just not for like the branching species, but also for the boulder corals. And we have been testing different materials. Here in the Bahamas, or like in this specific island, we are kind of isolated. We are in a remote location, so we're also trying to find other solutions besides the normal outplanting techniques or the normal substrates. So one of the things that we've been testing is, for example, conch shells. We have a lot of conch shells that they're like daily discard around the Bahamas for the fishermen. And then also some glass, like for example, glass bottles. So we have been testing if we can add these fragments to these kind of sustainable materials that at the same time will reduce a little bit the algae overgrowth and the sediment that normally is that is what attracts concrete substrates, for example. So right now we have 21 midwater nurseries, we have 8 raceways, we have been working with 10 coral species, and we've been propagating thousands of corals in our nurseries. So mostly the principle for these nurseries, we have these small fragments on the midwater structure. Then these corals grow, and after 1 year, we just put them back on the reef. And then once they're back on the reef, they will be able to reproduce and recover in a natural way. So this is a map of the Reef Rescue Network. So as you can see, there's a lot of, we have a lot of places in the Bahamas with coral nurseries. And the main idea behind this, the Reef Rescue Network, is that we just try to implement the same strategies and just try to keep track of the restoration process, just try more like data collection, and just keep track of the corals that they're being collected, outplanted. And then so we have more than 30 places with coral nurseries right now. So right now it's we also have some partners in San Lucia and Aruba, and the Reef Rescue Network is expanding. So back in 2019, we collaborate with Shed Aquarium. So we actually moved some corals from some of these nurseries. So just kind of bringing corals from different locations. So as I mentioned before, the Bahamas is really, really big. We have a lot of islands, and then also we have different conditions. So we actually move corals from different islands, and we had here corals from more than 5 different islands for this specific project. And then just for us, just to try to increase genetic diversity, we also work with sexual coral reproduction. And then for this, I don't know if you're familiar with SECOR, but SECOR, that actually stands for sexual coral reproduction. They are pioneers, and they have been working with sexual coral reproduction for years. They have really, really good technologies, really good just technologies, protocols that people can just follow, and they're just been expanding also in the Caribbean and worldwide to apply these techniques just to boost coral restoration, but based on sexual reproduction. So the substrate that you see in the top, that's like a design by SECOR. And we were really, really lucky that we were able to partner with them so we can also use these substrates. And then they also design CREEP, that is the coral rearing in situ basin. So in the case that people don't have the facilities on land, they can still do some coral spawning work in the water, just having this structure in the water, adding these substrates, and then adding like the baby corals in there so they can actually grow attached to these substrates, and then later on you can outplant them or just put them back on the reef. So back in 2016, Dr. Peterson from SECOR, he led a coral spawning workshop here in Cape Elutra Institute. A lot of people came here just to learn how to implement these techniques. And then after that, in 2019, we had a big coral restoration workshop here, also led by SECOR. We have a lot of partners coming here just to share these techniques, just to implement these kind of protocols for coral sexual reproduction. So after that, we started working here with this specific method, or with this specific coral reproduction strategy. So how this works, just in case you're not familiar with this, so we have some corals that they're broadcasters. That means that they release their gametes into the water column. They release the eggs and sperm. Then the fertilization happens in the water column. Then after fertilization, they go to embryogenesis, and then a larvae is created. And then this larvae is gonna find a spot in the reef, and then it's just gonna stay there and grow, and then just become like a larger coral eventually. So we have been working in coral spawning predictions for the Bahamas. So it's been super useful. There's other spawning predictions, especially from Curacao, that's like Southern Caribbean, but it changed a little bit since the coral spawning is related with moon cycles and sunset times. So we created these spawning predictions, and we divided the Bahamas in 3 zones. This is mostly because of the sunset time is a little bit different between the islands. We have been releasing these spawning predictions. So far, they've been accurate, and we will release the predictions for this year at the end of March, early April. So what we have been doing is that we go to the field, we collect these gametes, and then you see in the little tube, then it's like a mix of these gamete bundles or eggs and sperm. That's why the tube looks like a little white or a little cloudy. This is just the sperm. And then after fertilization, we have these swimming larvae. Then we have the recruits. And then later on, we are able to get some coral juveniles. This is all following Sea Corps protocols and advice, of course. This is one of the cribs that it was provided by Sea Corps, where we have all our substrates. So the idea is that we can have these in the water, have these crates full of substrates, then add the larvae, and then we just hope that the larvae think that's such a cool place where to grow, so they can attach to the substrate, and then they can just become little coral juvenile or coral recruit. Then after they are attached to a substrate, then we can move these substrates back, and we can plant them on the reef, or you can just like outplant them on the reef. So if you see that number on that picture, it's just because we do monitoring just to make sure. I mean, we will outplant thousands of these substrates. As you can see in the crib, there's like around 800. Of course, we are not going to monitor every single one, but we have like fixed transects where we can actually just go and check for, depending on the coral species, depending the year where they spawn, and just like have like a record of the growth and the survival of these corals on the reef, these new corals on the reef. This is just a couple of examples of some of the 2 species that we have been working with in a really successful way. So the first one is a brain coral, and the one at the bottom, that's a star coral. So this is just like a sequence on how they grew over time, and they're both attached to these Sea Corps substrates. They have like different shapes. One is a star, the other one is the tetrapod. So then you can actually see that the coral has been growing on top of the substrate. So this being like super successful. So far, we identify spawning peaks for 11 coral species. Of course, spawning peaks are being identified in other places in the Caribbean. There's more than 30 years of research in Curacao and the Southern Caribbean, but we didn't really have a lot of information of the Bahamas. So it was super important for us just to see spawning times, spawning days, just to see if it was like kind of similar with the Southern Caribbean. And so far, we raised millions of corals, and we outplanted like thousands of these substrates on the reef. And some of these species, they've been thriving after these massive bleaching events, which is really, really promising. I will talk a little bit more about that later. And then for the other way that the corals can reproduce, also with sexual coral reproduction, so we have brooders. So for this specific species, the fertilization is internal. The coral release the larvae, so pretty much this larvae is ready to go. And then this larvae is gonna just find a spot on the reef and then grew back and then just start the cycle again. So for this specific kind of corals, the cycles is like it had a lot of cycles per year, and they also become like an adult and can reproduce again like in a matter of 2 years, which is like easy or it's like easier for us if we want to study the whole reproduction cycle. So for this specific technique, we use our indoor facilities. So what we do is that we have, and this is also based on some protocols that Seacor use to collect these larvae. So we have some plastic containers, and then we have little collectors. So what we do is that every morning we check if there's any new larvae on these collectors. And then as you can see in the tube on the right, these larvae actually, they can glow in the dark. So it's just really easy to track with a UV light and a yellow filter. Then we collect the larvae, and then with this kind of larvae, we can just do some experimental work, or we can just like do some coral restoration work with these ones. And then at the bottom, you see like a little coral recruit. So after all these experiments, we also start working closely with Wageningen University in the Netherlands. So I'm doing my PhD with them as an external PhD candidate. So right now we're studying a lot of the early life stages of corals, also studying about this heat tolerance, the resilience, and most importantly, what are these trade-offs after the coral is being exposed to heat. So if they can survive, if they can, or if they can't, and then if they do, what happened after, like in terms of growth or in terms of competition. So we've been doing a lot of kind of like in the lab, so we can control the conditions, so we can control the water temperature, we can control the light, we can adjust like the light for a specific time for sunset, sunrise, moon cycle. And so we're able to get larvae from these corals just to conduct different kind of experiments, also just to see what is their swimming behavior, what is their feeding, if they can, after being exposed to heat, if they're still able to swim, if they're able to eat. So this is some experiments we've been conducting also with master students and PhD students from the Netherlands. So far, after we increase our capacity and we're able to do these experiments, we concluded so far 3 master theses with the students from the Netherlands. So this is going really well. But of course, after all these restoration efforts or outplanting back these corals, even if they came from sexual reproduction or asexual reproduction, it's important to know what happened with them. Also, it's important to know what happened after these bleaching events, what happened with these corals that were infected with a disease and they were treated. So we also do a lot of coral surveys just in these areas where we had these massive bleaching events, just to see what happened after, if we have any survivors that could mean that they're like more resilient than other coral species, and just try to shift a little bit efforts, just try to focus on those specific corals. And then for the treatment of the disease, just to see if the treatment work, if the coral survive or what happened with them. Then also for our outplanting sites. So right now we have 4 restoration sites where we move corals from our ocean-based nursery, and also from that they came from sexual coral reproduction. So we have our sites that we normally monitor just to try to see what is the survival and what is the growth. Then in the picture on the right, you can see that coral was infected, or like it had a stony coral tissue loss disease, and after the treatment, the disease completely stopped. So that coral actually survived. So this is just one example after treating thousands of corals just to see if the treatment is effective for the species. For our case, we had successful results with most of the corals that we treated, but some species, there's still a little bit tricky for them to recover, or maybe we just need a different technique, but that's kind of what we saw so far. Then also at the Perry Institute for Marine Science, we have a photogrammetry specialist. So he decided specific protocols just to implement this photogrammetry, just to go to our different reefs and just be able to take pictures of the whole reef, just to kind of see what is the status of the reef. If we had just not just for our outplanted corals, but also just to see natural reefs, what happened after these massive bleaching events, just to check mortality, diseases. So we set up like a specific plots or specific areas on the reef, and then we just take thousands of pictures, and then we just create like a specific model just to check what's happening with the reef. So then on the bottom right, you can see one of the markers. So just one of the corners for this specific plot. So then we just can go back and just take the exact same. It's like a permanent plot that we can just monitor over time just to see all these changes. In this picture, there's actually working with coral restoration could be a little bit challenging. It could be a little bit depressing, especially when you have these massive bleaching events and you have massive mortalities. But there is still a lot of hope, and that's why I have these two pictures here. So the picture on the left, that's a coral that we outplanted in one of our restoration sites. That was during the middle of the bleaching event. It's not the best picture, you can really see it, but that coral is actually pretty healthy. It didn't bleach, survive. The whole bleaching event was intact. Nothing happened to that coral. And that was actually pretty cool because that was one of the corals that we moved from Bimini. So that coral actually was not originally growing in this specific area of the Bahamas. It was a little bit more like close to the States. So that coral came from Bimini. In that specific restoration site, all our source colonies died, but the corals that are still there thriving and survive, they were corals that actually came from our ocean-based nurseries. So all the outplants that we have in that specific restoration site, they're thriving, which is really good. So we also had like a lot of mortality in our ocean-based nursery, yes, but the survivors, they're doing super great. And now in this specific place where all the source colonies or all the wild populations are gone, now we have new corals that they're like thriving. And then the picture on the right, so this coral is from it was sexually propagated. So this coral actually also survived the massive bleaching event, but the parents, or like the colonies that we used to collect the spawning back in 2021, all those corals died after the bleaching event, or also infected by stony coral tissue loss disease. So like all these generation that spawned before, most of these brain corals are gone now, but all these new corals, they're thriving. They did great during the bleaching event. So this is actually just pretty good. But then just besides the corals, we also realize is just in general, ecological restoration is super, super important for any like kind of restoration project. So we really wanted to start working with long spine sea urchins. So we start like a small project with some collectors, and the idea came pretty much because we had a research group here working with lobsters, and then they have lobster collectors to collect, let's say, just baby lobsters. And then in those collectors, they started to find some of these recruits or like these small sea urchins. So for the sea urchins, I don't know if you're familiar with that, but the long spine sea urchin or Darima antillarum, this species also had a massive mortality in the 80s. So long time ago, our shallow reefs, they used to have a lot of these sea urchins. These ones are super important grazers that will like prevent just the overgrowth of macroalgae. That's actually one big problem for corals right now, especially for coral juveniles and coral recruits. So we thought it was really important to started to study these long spine sea urchins. So with the support of Today and for Tomorrow Foundation, we started different like collectors, like testing different materials. And then when we were in the middle, or when we started with that specific research, we got in touch with other people working with sea urchins in the Caribbean. So there's a lot of studies going on in Ceiba, in Puerto Rico. So we joined the Caribbean-wide Darima settlement study last year, just installing these collectors with these bioballs that it was successful for other places. So we also joined this project. We installed 21 sea urchin collectors. We were able just to make 2 new collaborations. We also in Wageningen University, they've been working with rearing them in the lab. So just getting and also in Ceiba, just have with the adults and just be able just to get the gametes and rearing these sea urchins. So the idea for us this year is try to get this training and be able to apply that technique here. As far as I know, rearing sea urchins is being extremely challenging. Of course, we're not going to be the first ones trying that. There's a lot of people that tried before, but some people that they're behind successful results. So the idea is just to collaborate, try to share knowledge, and just like try to work together just to see if we can also rear these sea urchins. Then also here at the Cape Elutra Institute, so we're based at the Island School, we do a lot of outreach, education, and training that goes from visiting researchers. It could be PhD students, master students, interns, visitors, high school students, middle school students. So we have a lot of, we have like a really strong link with education. So far, we provided the Reef Rescue course. So with the Reef Rescue Network, there's a PADI specialty called the Reef Rescue course. So we provided so far 115 Reef Rescue certifications, where all these people got involved in coral restoration efforts, help with cleaning our nurseries, and help out planting the corals. We are also linked with a PADI dive center, so we also provide the aware PADI coral conservation specialty, mostly for non-divers, which is like great for example, for kids that they can, maybe younger than 10 years old, that they can dive yet, and around 241 dive certifications. So here at the Cape Elutra Institute, we have marine research and conservation internships. So for the marine interns, we also provide, besides of getting hands-on experience with research, they also get professional development. They can get certified, they can get advanced course, rescue, fish ID, oxygen provider, and then we also implementing a lot of different specialties with the Reef Rescue Network. With the they have like a new strategy just to involve more people with shark conservation, turtle conservation, all these with these different PADI specialties. So for our interns, there's always the option just to get training. Then we also work closely at the Island School. We have visiting programs. So this is pretty much students from different places. It could be high school students, university students that they come here to learn. So we provide a lot of lessons about like coral ID, coral conservation, also just get involved with the nursery and just help us to do some cleaning. And then also for them just to understand a little bit of the projects and the research that we are doing here. Then also at the Island School, we have the Young Leadership Program. So this is just young leaders from South Eleuthera, and they join this program. And then as part of the program, we help them to get open water certified. And then we just keep them for a little longer, just for them just to get more experience underwater and also just get involved with coral restoration efforts. So this is like a program that the Island School have, and it's just open for local local people here in the Lutra. Then as part of the semester program, we have high school students that they come here every year. So we normally do like a research experience for them. There's like they come here for 100 days. And we have been working with these students doing like a small research projects, but at the same time making sure that is like something impactful and useful for us. So small research projects like checking different, for example, in these pictures, measuring the growth of specific genotypes or testing conch shells, testing glass, just to see if the corals can grow on them or not. And they also learn about how the research is done, like what exactly, like how to read papers and how to write. And then at the end of the semester, they do a presentation and they also create a poster so they can share this knowledge and what they learned during the class and what they learned during the project with other students or with other people. So normally we do at the end of the semester a symposium. So we have a lot of people coming here just to see all these projects that the students have been working with. And most of the time is like real research, not just with the coral part, but like with the shark project, with the lobster team. So they just get involved in this research. And then with the Deep Creek Middle School, so these kids, this is like the local school here in South Eleuthera. They have like a really, really great opportunity to get SCUA certified. And then after their certification, we provide a lot of coral restoration training for them, just to learn about the coral nurseries, learn about the stony coral tissue loss disease. And then they actually explain to other people what they learn. They get like engaged with the project. They also have their presentations of learning when they explain other students that didn't have the chance to be involved in the same project. So they just share this knowledge with others. And then for the smaller kids, for grade 7, we provide lessons, just not about just coral ID, also just about fish ID. So why the reefs are so important, who lives there, like who, it's just not about the corals, but also about the fish, about the important species in the Bahamas, like the spiny lobster, the Naso grouper. And then we also they learn how to snorkel, they learn to identify corals. And we also do some interactive lessons with them just for them to learn, for example, about coral bleaching. So why some corals bleach, but other corals don't. And then with younger students, we just do different activities with them, also just to explain about coral reefs, just to take them out, just to check the reef by themselves, just do some drawing and coloring about different animals, or like what exactly is a coral polyp. So this year, the Bahamas Coral Innovation Hub is growing. We're gonna start working more closely with TNC again. So we just got a grant. So we're gonna improve our capacity. The idea is to increase collaborations and training. So this year, the idea is to expand and improve the lab that we have. So well, right now we're able to conduct a lot of experiments, but the idea is just to be able to do more or just improve for what we have. And then we're gonna have a really, really cool opportunity with Secor just to join training about the latest spawning techniques. So we're gonna do some training there. They have a lot of resources, a lot of protocols. And so it's just really important for us just to get that training. Ideally, we will get also some training for the sea urchins just to keep expanding and keep increasing our collaborations. And then we're gonna keep working closely with Wageningen University for the research side, just to keep bringing master students and PhD students and just keep working with different experiments for thermal resilience and heat tolerance. Also something really cool that is happening thanks to TNC is that we are going to be able to provide the marine research and conservation internship for Bahamians. So this is gonna be open for next semester, and then the idea is that we will have Bahamian interns working with us, which I'm really, really excited about because this is gonna be like fully funded. So we will be able to get more Bahamians involved with our project. Then we also gonna do more reef rescue course. So part of the reef rescue course is like a big part of this coral conservation and coral reef restoration efforts. And then we're gonna also just expand our educational materials. So far we have educational materials for different age groups, and the idea just to expand and just to create more, just for even if they can dive, but then at least they can understand the importance of coral reefs and what's happening with them and why it's so important to keep fighting and keep working to preserve coral reefs. For the research part, we're gonna keep working with this thermotolerance. We have different projects coming up, and of course, besides our normal work with the nursery, our normal work with the coral spawning, the idea just to keep doing more research, kind of like focus on this heat resilience. Also, we're gonna open up a new position here in South Lutra. So I'm also really excited about it. Probably we're gonna share about this new position pretty soon, maybe in the next week or two. And of course, all of this work is being possible all these years because we had a lot of partners, we had a lot of supporters, we have a lot of people that came here and help us to improve what we currently have. So it's just really important for us just to keep this knowledge exchange with different institutions. Of course, we are not the only ones working with coral restoration in the Bahamas or even in the Caribbean. So it's just so important just to exchange knowledge, help each other. So we've been doing a lot of good work thanks to our supporters and our partners. I don't know if you have any questions.
Thank you so much, Natalia. That was an awesome presentation. So before we do get into the Q&A session, I just want to remind everybody that we just dropped a webinar feedback form in the chat. So if you all can fill that out before you leave today or right after you leave today, that would be awesome. So Natalia, I'm just going to get started with the questions. And as we're asking and answering these questions, everybody, please feel free to send your questions in either the Q&A box or the chat box. But we're just going to use the next few minutes to answer your questions. So the first question is, other than shading, do you have or need a way to control water temperature in the outdoor raceways?
Yes, that's a great question. So actually for our outdoor raceways, we have a tank that is like a reservoir, and then we have a chiller connected to that tank. So then before the water goes into the outdoor raceways, the water goes first into this reservoir with the chiller, which is also like outside.
Okay, awesome. Okay, next question is, if we can get an e-certificate after this session.
Oh, that I don't know.
Okay, next is, are you using artificial salt water or deep salt water wells for your indoor raceways?
No, so actually we are lucky enough that the water quality here is really good. So as I mentioned before, we're in a really remote location. So we have a seawater pump that we just take the water from the ocean. So we are lucky enough for that, but I mean, still we have like a sand filter, we have US sterilizers, and then we also have other filtration systems, especially when we're working with coral spawning, just to make sure that the water quality is good. But no, it's natural seawater.
Okay, next question is, are there diving opportunities available for international citizens for collecting these samples at your institute in the Bahamas?
Yes, so we actually have so we are located at the Cape Elutra Island School, and there's a lot of opportunities, for example, for visiting researchers. It doesn't need to be like coral related work. Something, well, I didn't really mention that, but something cool about this place is that we have accommodation on site, we have boats, we have tanks, dive gear, we have like a lot of things, so people can come here and do their research. Then we also have the wet lab, um, and yeah, so the wet lab and the facilities. So we normally have a lot of visiting researchers coming here to do their own thing.
Okay, that's awesome to know. Next question is, do the sea core substrates require cement to attach to the reef, or do they just hold their place due to friction and their irregular shape?
So something really cool about the design of these substrates is because of that shape, you can just like wedge them on the reef, so you don't need cement for that. They just like, you can just like attach them, just like find like a crevice or something, then you can just like push it and it will stay there.
Oh, okay, awesome. Next question is, do you currently still use the crib system for most larval settlement aside from experiments within the land-based facility?
Um, It it depends. So for example, last year, so after the mass bleaching event, last year we didn't really have a lot of spawning happening. So it was not really worth it to use the creep because we had just like few spawning. So we actually did all the work in the lab. But that's also that I'm excited about, because during the our last C-Core Partner Symposium, they're gonna share like new technologies to use in a smaller scale than the creep. So that's, yeah, for that I will highly suggest to reach out to C-Core. They have like new technologies that we will learn, but we don't know yet.
Okay, next question is, as for your experiments with the university, aside from working with different temperatures, are you also experimenting with different acidity levels?
With acidity, like alkalinity?
Yes.
Uh, Not yet, but the idea is to start working more closely with alkalinity just to increase growth. But no, right now we're like kind of like in phase one, and but the idea it will be to start working. Yeah, it's just also for different alkalinity experiments that you need like CO2 tanks and like more controlled conditions. We don't have the capacity for that yet. It would be great, but yeah, we are still in a simple state.
Totally understood. Okay, next question is, does the research group use any cryopreservation tools?
No, also not yet. So, I don't know if you're familiar with, but we had some struggles with research permits, and like especially like exporting samples. It was like a little bit of a challenge. So that will be great. It's also in the horizon, but we don't have this specific technologies that we will need, or specific equipment. But no, not yet. But will be good. But no, we don't.
Okay. So, next question is coral restoration seems incredibly labor intensive and seems extremely difficult to scale. Have you given any consideration to automizing the process to produce tens of millions of coral polyps per year or per month?
Yeah, I agree. I agree. It's a lot of work, especially when you have these bleaching events and a lot of the work that you've been doing is gone. But I will say just to upscale, that's why we have been testing, like for example, different outplanting size just to see if we can outplant them sooner, for example. And definitely with coral spawning, that's like a great example of like upscaling because then you can produce millions of corals, like for example, using this creep, using these substrates, and then you add the larvae. So corals normally just release millions of these gametes into the water column, but the success afterwards is really low. So this kind of technique is just to try to improve or increase this success. So I will say for like just to increase numbers and upscale, like these methodologies used by C-Core are really good to apply.
Okay, awesome. Next question is, do you use plastic tie wraps to put the Secor substrate structures onto the reef?
No, we don't. So for those substrates, they just go on the reef without any zip tie, without any cement. There's a couple of like in some specific ones, if maybe we add like a zip tie, it's just to like a way for it's not just for all the outplants. Sometimes we use that for like a little tag, so we can keep track of which one is the one that we're monitoring over the years.
Okay, understood. Before we get to the last few set of questions, I just want to remind everybody once again to click the link in the chat. That'll take you to the webinar feedback form. We'd love it if you guys can fill that out. And if you have any more questions, now will be the time to drop them in the chat or the Q&A box. Before we get to the next question, just want to respond to Jay asking about the e-certificate after this webinar session. Unfortunately, no, we do not distribute e-certificates after the webinars. So apologies for that. But Natalia, next question is, any results on using conch shells for artificial reef structures?
They actually, yes, they seem to, well, we tried with the Stockholm coral and they grow on it. So we need to test more, but so far with that species, they grow on it. But so, with the conch shells, we've been testing like the two sides because like the outside is more like rugose and it gets with more sediment and like algae. So we've been testing also like the inside, which is like more smooth. So like it could be a little bit of a challenge first for them to attach or just to start growing on it because it's just so smooth. But the staghorn seems to like it.
Okay, thanks for that. And then last question for now is, hello, very practical question on the pictures of the sexual reproduction and settle larvae and growing coral on the substrate. We can see algae growth. Is this to be cleaned or is this coralline algae? And how can we see the difference?
One more time, sorry.
So I
Can you
think they're
repeat?
basically asking is the algae that we see like in the photos, are they going to be cleaned or are they like supposed to be there? They're a part of like the ecosystem.
Oh no, no, we don't clean it. So we normally just leave the coral. I'm gonna see if I can go back to that specific slide
Okay.
just to show you. Um, okay. This one, you mean? I guess.
Yes,
Eh,
I think that's the one they were referring to.
yeah, so for example, in this one, in the top one, like the first and the middle picture, there's no cleaning at all. We just let them there. We just let the coral just to grow, compete, and figure out. We don't really clean that because that's normally what is gonna happen when you outplant them on the reef. You just let them to survive and compete. The last picture, it just looks like a little nicer, but for that one, I just clean it to take the picture. But we normally don't. We just let them grow and compete. It's like the same for the bottom one. For the bottom one, there's no cleaning at all. And there's actually, you can see there's a little fire coral that also decided to grow there. But no, we just let them compete and survive.
Okay, wonderful. I don't think I see any more questions at the moment. So that is it for now. Thank you so much again, Natalia, for your presentation. And
Of
thank you
course.
all, everyone. And thank you all, everyone, for joining us today to learn more about the Bahamas Coral Innovation Hub, improving coral reef restoration through research, outreach, and education. Also, again, huge thank you to Natalia for this presentation. Very informative. Thank you so much. And for more information, please visit our website at theperryinstitute.org. Feel free to follow us on our social media channels at Perry Institute for Marine Science. This includes Facebook, Instagram, Twitter, and LinkedIn, soon to be TikTok as well. We hope you'll join us again for our future webinars. You can also find this webinar online in the near future. So keep an eye out for that in your emails. And thank you again, everybody. Please enjoy the rest of your day. Bye, everyone.
Thank you.